Hello and welcome to the GCSE Biology tutorial for topic B1 Cell Biology. This is part 2, so covers cell division and transport in cells. Starting off with cell division. To understand this, we need to know a little bit more about what's actually happening inside a cell. So inside a cell you have the nucleus, and we know that it contains genetic material. Genetic material is inside chromosomes. In normal body cells of a human, there's 46 chromosomes, or 23 pairs, and in sex cells, like sperm and egg of humans, there's 23. Chromosomes are made up of genes, and genes are made of DNA. When a cell grows and multiplies, it does so during a process called mitosis, and mitosis is normally for growth and repair, and it's only one part of a cell growth cycle. You can see here, for a cell to grow, the DNA has to duplicate, then mitosis happens, and then the cells split. Let's look at mitosis in a little bit more detail. So here's a chromosome which then replicates. The DNA has to copy. This is really important. If the DNA doesn't copy, the cells won't be identical. Then the cell can begin to divide. Once the two chromosomes of each copy are formed, the nuclear membrane breaks down, the chromosomes line up in the centre of the cell, and then they are pulled apart to either side of the cell by spindle fibres. Once they're pulled apart, they then split and form two identical cells. Those two cells are identical to each other, but they're also identical to the cell you started with. So that is the process of mitosis. Mitosis also happens in plants, in particular in the area called the meristem, which is the tip of the plant, and other parts of the plant also differentiate and specialise, just like animal cells. Prokaryotic cells also multiply and divide. Most bacteria cells can divide every 20 minutes, so in 20 minutes, 1 will become 2, in 42 will become 4, and 8, and 16, and so on. Now, this type of cell division can occur in bacteria because they only have one cell, so they don't have a complicated structure, so this is also how they reproduce, and it's called binary fission. If you look at the diagram, you can see that before the bacteria splits, the chromosomal DNA in the plasmid also has to copy, otherwise you won't get cells that have identical features. Now, the number of plasmids might vary, but the chromosomal DNA has to copy and split before each divide. Moving on to stem cells. Stem cells are unspecialised cells that have the ability to become any kind of cell, so they're really powerful. And there's two types. There's embryonic stem cells, which come from embryos, and they're more powerful because they could form any kind of cell. And adult stem cells come from cells inside adult tissue, but they will only become the cells they're taken from. So say, for example, you take cells from the bone marrow, they will only be able to make bone marrow cells. Now, you need to know about the reasons for and against stem cell research, and we'll look at that more in the 6-mark question. This diagram shows how stem cells can be used. You would need to get a cell from a patient that needs some more cells. So they might need some more tissue, or they might have a genetic disease, or there might be some part of their cells that they need to regenerate. You take the nucleus from the patient's cell and transfer it into an empty donor egg cell. And then you stimulate that with electricity, and the egg starts to divide, and it's forming an embryo. But at this point then you stop it, and you take the stem cells out at this point, because these cells then can become the cells that you want. You can manipulate them to become the cells that you want. And they can then be used for the patient, and the patient won't reject them because they're from their own body, their nucleus, so it's the same as their cells. And that's called therapeutic use. You also need to know about transport in cells. 3 processes: diffusion, osmosis, and active transport. Diffusion is a movement of particles from high to low concentration, and the larger the surface area, the faster diffusion will happen. We'll look at some examples of that later. Osmosis is the movement of water across a partially permeable membrane, which means a membrane that allows some substances through and others not. And that's a movement of water from a high concentration of water to a low concentration of water. We'll look at that in the practical. And active transport, that's the opposite of diffusion, so it's a movement of particles from low to high concentration. Examples of that are root hair cells. They use active transport to get minerals from soil, even when there's a low concentration in the soil compared to in the plant. It also occurs in your small intestine to absorb glucose. Now, active transport requires energy. So, going back to the practical with osmosis, this practical includes using potato tubes. So the first thing you would do is you cut up potatoes into tubes with the same mass and size, and you put those tubes into different solutions. So one of just pure water, and then different concentrations of sugar solutions, so 20% sugar, 40% sugar, and so on. Before you place them in, you'll measure the mass of the potatoes and then leave them for about 24 hours. Take them out, dry them, and re-weigh them. If the potatoes have got heavier, they've taken in water. If they have got lighter in mass, the water has been taken out by a process called osmosis. You can then also calculate the percentage change in mass. So the dependent variable is the mass of the potato tube. The independent variable, the thing that you're changing, is the concentration of the sugar. You can then find out at what concentrations the potato gained or lost water. In general, you would find that when it's a higher concentration of sugar, the potato will lose weight because water will leave it. And then when it's a more watery solution, the potato will get heavier because water will move into it. And that's the process of osmosis. You'll also need to know about a practical looking at antibiotics and bacterial growth. To do this, you would need agar plates. Agar plates are dishes full of nutrient jelly, which are a really encouraging place for bacteria to grow. You will have seen one of these in class. Now you're trying to see which antibiotics are the best at working. So you have different antibiotics. In this picture, you've got 3 different antibiotic samples and a control. The control is no antibiotic at all, but just something to use as a comparison. So that would be just soaked in water. So each of these antibiotics have been soaked in each of these samples have been soaked in different antibiotics. You will then put this on an agar dish and cover it with a bacteria, and then leave it for 48 hours in an incubator about 25 degrees. After 48 hours, you would examine the dish, and the antibiotics that's the most effective will be the one that's got the bigger inhibition zone, which means the whiter circle here is bigger. If that zone's bigger, it means the bacteria has been killed by the antibiotic. The bigger the inhibition zone, the more bacteria has been killed. And you can actually calculate the area of the circle as well by using the calculation area equals pi r squared. Now, points to note, when you are carrying out practicals using bacteria, you have to make sure you use sterile techniques. I'll just go through them. First of all, the agar jelly. That is a jelly liquid that is the medium for the bacteria to grow in. You have to use a sterile wire when transferring bacteria to the agar, and this can be sterilised by putting it into a Bunsen burner flame until it goes bright red. You also need to sterilise everything beforehand to kill any unwanted microbes, because you only want to culture the bacteria in the practical. You store the dish upside down to avoid condensation, and you don't open it once you've started to culture the bacteria inside. And once you finish the practical, you wouldn't throw this in the bin, you put it in something called an autoclave, and that destroys all the bacteria. So anytime pupils or scientists are working with bacteria or microbes, you have to follow aseptic and sterile techniques. Okay, if you pause the tutorial now and try and answer these questions. So what are stem cells? They're unspecialised cells that have the ability to differentiate and become any kind of cell. 2 differences between diffusion and active transport. Diffusion is the movement of particles from high to low concentration, but active transport is the movement of particles from low to high concentration, and requires energy. Where could diffusion happen in the body? Well, it could happen in the lungs, in the alveoli, and in the small intestine in the villi. We'll look at those in a little bit more detail next. What happens to the potato tubes in water and why? They get heavier, they get increase in mass because water moves into them via osmosis. So, organisms need exchange surfaces to take in what they need and get rid of waste. So in other words, organisms don't exist as just one cell. They're made of many cells. So they need an area to be able to get things in, like oxygen, and remove things like carbon dioxide. Now the problem is, the larger the organism, the smaller its surface area to volume ratio. So you can see here, a larger cube has a smaller surface area to volume ratio than a small cube. So this means that organisms need to have exchange surfaces adapted to be able to take in and remove substances. So let's go through some examples that you need to know. In the lungs, you have alveoli, and these are found inside the lungs and are millions and millions of air sacs. And you can see one alveoli just above in the diagram. Now, when you breathe, oxygen goes into the lungs and into the alveoli, and then through the alveoli into the blood. And they does that through diffusion. So for this to happen, the alveoli have to have a very thin lining and a thin wall and a good supply of blood around them. So as soon as the oxygen moves out, it goes into the blood. At the same time, carbon dioxide also leaves the blood and moves into the alveoli, so you can breathe it out. So the alveoli are adapted and allow for diffusion of oxygen and carbon dioxide in the lungs. There's also important exchange surfaces in a plant. So this is a cross-section of a leaf. The underside of a leaf actually has holes called stomata, and they allow oxygen and water vapour to diffuse out and carbon dioxide to move in. And they're controlled by guard cells. We'll look more at that in the plants topic. They also have a flat shape and they have lots of air spaces inside the leaf to increase diffusion rates. Villi. Villi are found in the small intestine, and they have allow the small intestine to have a large surface area so the digested food like glucose can be absorbed into the blood. Again, like alveoli, they're very thin, they're a single layer of cells and they've got a good supply of blood around them. Another example of exchange surfaces is in fish. So fish have large surface area for gas exchange on their gills. Water containing oxygen moves through the mouth and then passes out of the gills. As that happens, oxygen goes into the blood through diffusion and carbon dioxide comes out. If you go looking closer at a gill, the gills are made of gill filaments which make the surface area even bigger. And again, they're covered in blood and they're made of a thin layer. So there's a common thing here. To make diffusion happen faster, it needs to have a good supply of blood nearby for the substances to move in and out of, and a thin layer of cells for diffusion to happen. So here's a 6-mark question. If you pause the tutorial and try this. Okay, first of all, you need to say what stem cells are. So remember, they have the ability to differentiate and specialise into many other kinds of cells. But what can they be used for? Well, stem cells could be used for research to treat diseases like diabetes for people who don't have the insulin gene, tissue repair, so repairing certain cells, and nerve cell growth. They can also help us understand how cells grow and can be used for cloning of cells and tissues of a person's body without them rejecting it. They can also be used to make plants that are cloned cheaply and quickly. But there is problems with stem cells. Adult stem cells don't differentiate into all types of cells, so they're not as varied, they're more specific. Embryonic stem cells do, but that involves the destruction of embryos. And destroying embryos is unethical because that is seen as life. So there is ethical and religious objections to this. Also, by passing cells from one organism to another, there is a risk of a viral infection being passed on as well. So with this answer, try not to be vague and think of some examples of how stem cells can be used, for example, in helping people with bone marrow disease or diabetes. Okay, we have covered mitosis, stem cells, diffusion, osmosis, active transport and exchange surfaces, and looked at 2 practicals. Thank you.